Hello everyone, this is the Theoretical Doctor and welcome back to my channel. If you're new here, welcome. This is a series on the chapter Immunity on chapter 6 of the STPM Semester 2 Biology series. Check out the full playlist on my channel. If you want access to flashcards on this topic and other videos, do click the link below. It is available on my Pinterest and slides on this topic are available on my website. In this video, we will be continuing on humoral or antibody-mediated immune response, whereby we will dive into the response of B cells with an infection through the humoral immune response with and without helper T cells, as well as the primary and secondary immune response. So the humoral response results in the synthesis and secretion of the antibodies of B lymphocytes. It may or may not require the participation of T4 cells, which are the helper T cells. So first, I will talk about if helper T cells are involved. Helper T cells are also involved in the cell-mediated immune response, which was in my previous video on cell-mediated immune response. So for more explanation on that, do check that out. Just like in cell-mediated immune response, when the antigen is being displayed on class 2 MHC proteins, the helper T cells are being stimulated. So since MHC proteins are the same, which are class 2, the helper T cell sees it and binds to it with the help of CD4. Okay, so let's get into this. So when a B cell binds to the antigens of the pathogen is then taken into B cell by receptor-mediated endocytosis and digested. The antigen then binds with the class 2 MHC protein. MHC stands for major histocompatibility and moves to the cell surface. So now the B cell becomes an antigen presenting cell and presents the same antigen that fits in the receptor of T4. So this part here, as you can see, is very similar to the cell-mediated immune response when helper T cells are involved. So the T4 or helper T cell then binds to the APC and begins to secrete interleukin-2. This interleukin-2 will activate the B cell. So this part here differs. Now we're going to talk about what happens to the B cell, not what happens to the T cell, but the B. So the activated B cell then divides repeatedly into plasma cells and memory B cells. So these plasma cells will produce and secrete large quantities of antibodies that are specific to the original antigen. So it has to be specific because it has to fit to the epitope of that antigen. So in this response, both T4 cells and B cells are identified and selected by the antigen. This is a kind of humoral immune response to a T-dependent antigen. T4 cells are involved here. T4 cells are the helper T cells as well as the B cells. So the first part is very similar to cell-mediated immune response with the helper T cells. But if they're talking about humoral response, you have to tell what is going to happen to the B cells. So this is a diagrammatic representation of the humoral antibody-mediated immune response. You can pause the video here and refer back to the explanation to help you better understand without the helper T cells now. So what happens? Because humoral antibody-mediated immune response can function with or without the helper T cells. Here is without. So this is where the memory B cells plays the most important role, because not all the time the helper T cells will come into play. Like in this case, what if helper T cells are not there? Will the B cells still work? Yes. So the B cells that has recognized the antigen will successfully bind to it, and then the B cell is stimulated to proliferate, to divide into clones of plasma cells and memory B cells. So these plasma cells, like I mentioned earlier, they'll produce large amount of antibodies that are specific to the antigen, meaning the epitope of that antigen. It has to fit like lock and key. So this process of antigen selected proliferation is known as clonal selection. As you can see over here, the antigen comes and it already recognizes the antigen and it will bind. Okay, it'll bind to the epitope of the antigen, and then this B cell will be stimulated to form more memory cells and more plasma cells. cells. Primary and secondary immune response. So when lymphocytes are first exposed to a particular antigen, keyword here is first exposed, the primary immune response is generated, and the important antibody here is IgM or immunoglobulin M. This will take about 10 to 17 days to produce maximum effector response, meaning to secrete the antibodies to attack that. So it's going to take some time because it's the first time the infection occurs. So during this time, the individual is likely to show symptoms of the disease. Now, if the same infection comes again, if the individual is subsequently exposed to the same antigen again and again and again, it's no longer primary immune response because memory cells have been formed. They will recognize it. So the secondary immune response is produced. So the immunoglobulin important here is IgG or immunoglobulin G. So this is a much faster and stronger response. The ability to generate secondary immune response is due to the production of memory cells, both B cells and T cells. So if you can see in the graph over here, during the first exposure, the primary immune response comes into play. But now if the second time the infection occurs, the secondary immune response will come into play, and they are more faster and more stronger. So if you want more explanation on the different types of immunoglobulins, their structure as well as the shape, do check out my previous video on the human lymphatic system and function, plus the structure and roles of antibody, antigen, and defense cells, where I elaborated more on this. If you've reached this point, thank you very much. Hit the like button if you found it helpful, as it does help in reaching out to others as well. Do support my channel by pressing the subscribe button so I will know, and it gives me motivation to continue producing content like this in the future. And finally, share this video to those who might find this helpful. See you in the next one.